Forgiving my voice. Today I will talk about methods of bioabsorption and bioleaching by microbial genera, not by algae or by photocatalyst, but by bioabsorption and bioleaching by microbial genera and microorganisms. The work is based on article published in 2022 in Sustainability by Professor Prithviman Kaur from Punjab Agricultural University in India, entitled Bioabsorption and Bioleaching of Heavy Metals from Electronic Waste Varied with Microbial Genera. The electronics, after use, generates much waste because besides silicons, it contains much different metals, heavy like gold, copper, and rare metals. Thus can become toxic for if wrongly recycled, and which are excavating and mining produce much of waste. Therefore, here as a method, it's important to find method to produce metal photocatalyst also, because as we see, the photocatalytical the composition to produce biofuels. One option is using metals, but this option where to find, and one is the recovering of metals from already the electronic waste. And one option beside algae absorption and is using a pseudomonas as bacteria, which can be also used for water splitting. But in this case, Professor Prithviman Kaur developed this bacteria for using the lactase enzyme and using the absorption of copper and iron metals from air waste. This case is using many waste discarded electronic products, printers, photocopiers, refrigerators, mobile sets, phones. All these are coming big waste, and unfortunately, rich countries often abuses poor countries like Congo or Africa, and even Poland now, to split from European countries to poor countries to landfilling, and this makes big harms to the environment, fakely utilizing it in Europe. So there is a big fake producing if not finding proper method. This method is also very promising for these countries, because if we give this to these African countries, They can organize this waste to produce recyclability, not to toxic the land and make the big trouble for local communities. Are highly toxic, some are non-degradable, and living and use and living dumping and big troubles in for local communities, cancerogenic and destroying biological organisms. And bioabsorption and bioleaching is one of the main strategies that can absorb and recover. There is also macroanalysm, and there can be also some besides, also different bacteria and fungi, and even some uh, nano, um, some some organisms like fungi, like Aspergillus, Pseudomonas, and some supernatant culture. There is also the microbes requiring lacas as proper multi-couple organism involved. There is also can oxidize phenolic compounds, and these fungi are more efficient because already more bigger lacase enzyme. This lacase enzyme is also important to treat, for example, treat the cellulose and lignin. So we see that this fungi, besides using as lignin and uh, lignin uh, separation from cellulose waste, they can also be sites using, they can detoxify absorb hard metals, and thus two functions uh, of this fungi, the cultivation. There is also by this professor develops also some for these cases nematodes, but in this research he don't show nematodes that can also eat this and therefore absorbing metals. But here micro use is 7 microcarpia cultures by fungi and pseudomonas from bacteria and fungi compared. Preparation uh, was using there's some element of printed circuit boards were used as the feed for this bacteria and organ and fungi. So 5 grams of PCB was washed in distilled water and followed by ethanol washing and then placed in liquid minimal broth, mushroom or for fungal and bacteria minimal or compounds with some glucose as and asparagine as nutrient initial and starting material, and some magnesium sulfate and phosphate, dipotassium phosphate as salt addition. Therefore, we see that in case here we can use, as mentioned, some wastewater to may have this phosphate. So, in well, good pain can be used also with some wastewater for where this magnesium phosphate is already. The lacase activity was inoculated with different and rotary shake controlled bar was used. And wait after 4 days. The bioabsorption by this was assayed after 8 to 20 days. And this shows both was inoculated and leaching was after these 20 days. Checking how much leached metals was made by the solution of leach was checking in aqua regia solution. So chloride with nitric acid, and the certification and such was checking the bioabsorption efficiency. Copper rate was calculated, and how was the using? As the results were used, how much of PCB was used, and in case of using the circuit base has several metals: copper, zinc, nickel, iron, cadmium, gold, silver, cobaltium, lead, and palladium. And in Kison's study, the using was up to, in case of iron, using this fungi with or bacterial was much higher than other earlier mentions, while in case of copper was only beside using the chemical and biological waste pretreatment from mobile phones was better using. So here we see that was already that for mobile phones, similar method was even better than in case of polymer circuit. And so, but here polymer the the printed circuit boards was better. And this, the lacase activity was highest in case of Pleurotus florida, so in case of bacteria, that in case of fungi. So we see that fungi are between 20 are similar in efficiency, but after 20 days, it was between 16 and 24 days. The activity of Florida bacteria was much higher than in case of which is also fungi, but microbial fungi was much the highest. The bacteria was much smaller activity. So mostly some pseudomonas showing that this fungi was much higher. In case of this Pleurotus florida, the bioleaching and bioabsorption of metals, copper and iron was much better, or smartly better than Pseudomonas, only to 2% higher than Pseudomonas. So Pseudomonas, besides being a bacteria that can split ammonia, they can also use as copper recovery and bioabsorption. In case of iron, the In case of iron, even bacteria were better than fungi. So if we want to recover the waste, electronic waste from iron, and absorb the bacteria are seriously better than fungi. But in case of copper, it is different. So deciding we need to develop method both using fungi and bacteria, which are also can be used in other ways due to the splitting possibilities. Because this showing that these fungi, which are good for cellulose or lignin, the composition, can also split by bioabsorption of heavy metals, while the bacteria that can split some ammonia into hydrogen, they can be also used as desorption, especially of iron, metals, and recovery. So we see that there's some fungi that are already can be used as for lignin and lignocellulose separation. So important to have cellulose high efficiency of for biofuels. Then can be also used as biosorption biology. Of course, the similar as bacteria has algas, but sometimes algas are not fine, or microalgas. However, microalgas can give also capturing oxygen. But here we see that in some cases, when we want to do ignite, for example, where carbon dioxide is unobtainable, or in case the algas are in region that so that for example, which are not familiar to the algas, for example, in winter, hardly winter, maybe such design is much better. So thank you very much for listening me.